So let's move on to a different kind of bacteria. We have cyanobacteria. So this is often called blue-green algae because it often presents as filamentous algae, but they are still single-celled bacteria. They just grow colonial in these long filamentous chains occasionally. They are the only bacteria with chloroplasts, and they need to be in moist environments. So when you find them in the soil, you find them in wetter soils. They are famous for a few reasons. One, because they can cause algal blooms that can be toxic, but two, because they are probably where the great oxygenation event happened. Was cyanobacteria. Oxygen didn't used to be common on our planet. They think that the proliferation of cyanobacteria created oxygen. When you look through our geologic record, you can see where rocks all of a sudden are rusty, that iron in those rocks has been oxidized. And prior to that, there wasn't oxygen to do that. That there was some kind of switch in the atmospheric makeup where oxygen became plentiful, and they think it's cyanobacteria. They also think that chloroplasts in plants are probably some ancient form of cyanobacteria that became mutualistic with a vascular plant and eventually got trapped inside a plant. Kind of like how mitochondria were probably their own cell, they have their own DNA. Chloroplasts, same idea, probably are trapped bacteria. When we find them in the soil, they often are on very wet soils, and they'll create like a crust on the surface because they are photosynthesizing. So they need that solar radiation to photosynthesize. So you're not finding them deep in the soil. You'll find them on the surface. And if that wet soil then dries out, it'll help create this crust on the surface, which does help some of these very wet soils that have these drought periods. It does help them have more stability. So it's thought to help with erosion control. So you might have seen soils with these cyanobacteria crusts. Now this is more important for when we actually go out and sample. Typically, I set up sites where I have some students sampling soils from the field across the street that is abandoned agricultural land, and I have some students sampling from the forest behind the campus, and we can compare how much bacteria and fungus and other critters like arthropods and earthworms and nematodes that we're going to find in those different soils. The amount of fungus is just exponential between an agricultural land where the soils are disturbed and often a neutral pH compared to a conifer forest where you have a undisturbed acidic soil. You get a lot more funguses. It's crazy. No matter where you are, you're going to have a ton of bacteria. And we usually extrapolate and find out exactly how many bacteria we have per 1 milliliter of soil and how that extrapolates to 1 hectare of forest. It's ridiculous how much bacteria and fungi are in the soil. But the number, like we talked about arthropods, the number of mites we find in the soil is also astronomical. Nematodes are a little bit harder to find. They're there, just in our microscope techniques, sometimes you don't always see them because they're like see-through. But amazing, the numbers of once you start looking in the soil with a microscope, the things that are in there, how alive it is. Lastly, I want to talk about Archaea very shortly, purely because a lot of you are coming from backgrounds where you might not take any microbiology, But we do have archaea in the soil as well sometimes. We used to think of archaea as an extreme bacteria. These were bacteria that could grow in extreme environments. We now realize that archaea have a lot more traits more similar to eukaryotes than they do to bacteria, but have adapted in a way that they can take advantage to those extreme environments. So most of them do live in environments where bacteria and eukaryotes cannot thrive. So like a famous one are halophiles that live in salt water that nothing else could live in. But we also see them in extreme temperatures, in hot water springs. Yeah, all over the place. I'm filming this at home and there's a pair of vireos in the tree and they're distracting me. I'm sorry, guys. Okay, sorry. Yeah, you've probably seen pictures of these colorful hot springs. That's because of extremophiles. I know when I was a kid, I read about them in National Geographic magazines. Pretty famous. But new research is showing more information about them. So we thought that they were only in these harsh environments, but as we're learning more genetically about archaea, we're finding that we can find them in soil environments, especially soil environments that are extreme. So soils that are toxic or have high levels of minerals, for example, old mining sites. We're finding them in anaerobic soils and super salty soils. And recently there was a paper talking about Cranarchaeota, which is one of the archaic organisms that is possibly an important nitrogen cycler in a lot of the soils that we have. And last year I was lucky enough to talk to a professor at Laurentian who was looking at bacteria and archaea and their ability to digest toxic metals in the soils on the mining sites in Sudbury. So there's a lot of use for these organisms in soils that we're only just learning about that wouldn't have been in my soil science class when I took it in the early 2000s. We're learning pretty quickly more and more about archaea. So before we end this PowerPoint presentation, I wanted to add a slide on Phaeroplasma acidophilium. It's just one of many examples of archaea that you can find in the soils in Northern Ontario. So this particular one is found in the mine tailings in Sudbury. It is iron oxidizing, so it makes a red color because it's turning the iron in the soil red, rusty. It loves acid environments, which is typical of our archaea, how they're extremophiles. And I love this word, it's chemolithic autotrophic. So this means chemo uses chemicals, lithic rocks, auto makes its own trophic food. So chemolithic autotrophic are organisms that can use chemicals to get the nutrients they need from the rocks. So just like we're breathing in oxygen and eating carbon, so we're eating plants. Plants are taking carbon from the atmosphere, from carbon dioxide, and we're getting carbon from those plants. Autotrophs versus heterotrophs. These guys are getting carbon from the rocks. So it's pretty neat. And there's lots of different Phaeroplasma that are mineral oxidizing, just not necessarily iron oxidizing. But this one is found in Sudbury frequently. And last winter we had the pleasure of Dr. Nadia Nikitschuk coming to our lovely town and speaking, and a bunch of biology students went and saw her speak. And she does research on bacteria and archaea, and specifically looking at ones that are eating minerals. So I included one of her more recent publications here, but she's a pretty cool lady. And yeah, that will be the end of our soil organisms. I'll see you guys soon.